Well, thank you very much for that introduction, and thank you as well very much to the AMMF for the invitation to speak. It's always a huge pleasure to be here. Now, I'm going to talk to you over the next 20 minutes about molecular profiling in biliary tract cancers, but what I'm not going to do here is focus in on the intricacies of the technologies. There is, of course, going to be a little bit of that, but what I'm really going to focus in on is the issue of equity and making sure that wherever you live in the UK, you can access the same type of testing. I'm going to highlight areas where that is the case, and that is a phenomenal achievement, and areas where there's still work to be done. So, as I said, you're not going to avoid discussion of some of the technicalities here. So I'm going to start by laying out the basics by stating what I'm sure pretty much all of you know already, which is why is molecular profiling so important in this family of diseases. Then I am going to talk a little bit about the technical aspects, talking about what different ways of testing there are. And that is then going to build up to the final few minutes where I'm going to talk about how that testing is delivered across the 4 nations of the UK at the moment. And as I say, where things are equal in terms of access and where I think there's still room for development. So starting from the top, why does molecular profiling in this group of cancers matter? And I know that pretty much everybody in this room knows this. Fundamentally, the main reason is that we're not talking about a single disease here. Even within cholangiocarcinoma, I think it's fair to say that nowadays this is a family of related diseases rather than one single diagnosis. And those different diseases are characterized by different molecular alterations. You look at this, it kind of looks academic, but it does have real-life clinical impact because these different molecular subtypes are associated with different levels of disease aggressiveness, and very importantly, patients who fall into these different categories have better or poorer responses to certain drugs. This is the whole idea of personalized cancer therapy. These 5, I think it's fair to say, are the standard molecular groups that we deal with in the UK at the moment, but this is a rapidly growing family as we make finer and finer distinctions between these different diseases. So fundamentally, the reason this matters is that it allows us to personalize treatment. But something which I think is less recognized, but equally important, is that, as we've already heard, cholangiocarcinoma is a difficult cancer to diagnose, both in terms of picking it up clinically, but also for us as pathologists. It's a really tricky diagnosis to make. And I want to make the argument that early molecular testing not only gets patients access to the right care, it also helps us for the first time to positively make the diagnosis of cholangiocarcinoma, which is clearly the first step before everything else comes. So what do I mean by that? Well, and I promise, these are the only few pathology images in the whole slide deck. That is a picture of a cholangiocarcinoma. But that could equally well be a lung cancer, a breast cancer, a gastric cancer. There are a few differences, but fundamentally, these things look the same down the microscope. There's nothing really in that says, yes, this is definitely cholangiocarcinoma. So the tool that we as pathologists reach for in situations like that, where we can't say where a cancer is coming from, is immunohistochemistry. That's a way of looking at what molecules the tumor cells are expressing. Now, for lung, we've got a really, really good marker, TTF1. If that's positive, you're probably dealing with a lung cancer. For gynecological cancers, you can tell which cancers I report from the list I've got here. For gynecological cancers, Paxate is a really, really good marker. Not totally specific, but a very good marker. There is no marker, though, for biliary tract cancers. There's no marker for cholangiocarcinoma. So the upshot of this is that conventional pathological tools cannot make a positive diagnosis of cholangiocarcinoma. It's essentially a diagnosis of exclusion, and that's problematic. But what is specific in this context is the molecular alterations. Because if you have a cancer which looks down the microscope like what I'm showing you at the top, and you detect an FGFR2 fusion, I think you can quite easily make the diagnosis of cholangiocarcinoma. Likewise, IDH1 variants. None of these things are foolproof, but it gives you very strong evidence. And in fact, the only evidence that a pathologist has to say with a high degree of certainty, this is cholangiocarcinoma. You can only do that, though, if you test, and if you test early on in the diagnostic pathway. So, that's why it's important. What I'm going to move on to talk about now is how we actually do the testing. And I should start with an apology, because the analogy which I'm going to give is going to horribly patronize, I'm sure, most of you, but bear with me, because I think it does exemplify it nicely. Let's imagine that you decide that you want to cook a meal. You go online, Google, find a load of recipes on a website. That's kind of the equivalent of your DNA. Your DNA is a big recipe book containing all of the recipes that make you. You find a recipe that you like and you decide you're going to print that off. That's the equivalent of RNA. RNA is one single recipe which has been printed from that big recipe book. And then you go away, go to the hob, cook your meal, you end up with a meal. That is your protein. It's the final end product. It's the thing that matters from this entire process. And this is called the central dogma of molecular biology. You have your recipe book in the DNA, you have individual recipes expressed as RNA, and then that is converted to protein, which is what actually does what needs to be done. And the reason I'm laboring this point is that this illustrates an important dichotomy in the way we do molecular testing, because you can look for alterations at the level of DNA or RNA, what we call genomic biomarkers, or you can look for them at the level of protein, what we call proteomic biomarkers. Particular alterations may be detectable only at the genomic level or only at the proteomic level. But because you can have an alteration at the DNA level, leading to an alteration at the RNA level, leading to an alteration at the protein level, you potentially have choices for some biomarkers. You can detect them either genomically or proteomically, and there are reasons you might choose one or the other. And I should say that genomic testing, rightly, gets a lot of attention, but I have to say proteomic testing has been the poor relation in all of this, even though actually most of the new biomarkers we've seen in the last few years have been proteomic. So how does this apply in biliary tract cancer, and specifically in cholangiocarcinoma? Well, you have some alterations which are purely genomic. These are IDH1, FGFR2. You don't really have an option there. Again, NTRK, that's pretty much only genomic. But then you come on to some where you do genuinely have a decision to make. So mismatch repair or microsatellite instability status, you have a choice. They're two different ways of looking at the same thing. Likewise, for HER2, you can either look at it at the genomic level or at the proteomic level. And just to emphasize again, there are lots more things coming, and they too have this decision to make. And I'm going to come on to how we make that decision in a few minutes. But first of all, what I'm going to do is lay out the general approaches that we use to detect genomic alterations and proteomic alterations, before I then tell you which I think is the best one to go for in each scenario. So starting with genomic testing, how can we deliver this? Well, the approach which we've historically used is what we call a single gene approach. So you use one test to look at one gene. If you want to look at 2 genes, you have to do 2 tests, 5 genes, 5 tests, etc. And you can immediately see the issue here is that this becomes very inefficient when you're looking at large numbers of genes. Having said that, this does have a role still. These tests can be very, very quick. They can work in really, really tiny samples that you couldn't do anything else on. So they still do have a role. But I think it's fair to say that in the last few years, they have been to at least some extent replaced by the new approach to testing, which is next generation sequencing, or NGS. And the fundamental idea with NGS is that you use a single test to look at lots and lots of targets at once. So you can immediately see why this is a much more efficient approach where you have to look at lots of targets. So for example, in a cholangiocarcinoma, you would do a DNA NGS panel to look at IDH1, as well as probably quite a lot of other genes as well. And you would use an RNA panel to look at things like FGFR2 and NTRK, as well as lots and lots of other things at the same time. Clearly, that's going to be more efficient if you're looking at lots of genes, which we're now doing in cholangiocarcinoma. And as I say, the Genomic Medicine Service in England has really driven a movement away from 1 and towards 2 in the last few years, which is a very good thing. So NGS. We talk about NGS like it's a thing. It isn't really. It's really just a loose collection of technologies which only really have in common the fact that they look at lots of different things at once. And there's variability in the scope of it. So at one end of the spectrum, you can do whole genome sequencing, where you look at the entire genome. You will, in theory, detect absolutely anything which is going on in the genome of that cancer. You could do whole exome sequencing, where you only look at the bits of the DNA which encode proteins. Still very, very extensive, but not quite as much as whole genome. But what we tend to do in routine practice is NGS panel testing, where instead of just looking at big chunks of the genome, you look at particular genes which you're interested in. And those panels can cover hundreds of genes. They can cover maybe up to 100 genes. they could cover only a few dozen genes. And you may well be thinking, looking at this, well, why would you bother with these piddly little DNA NGS panels when you're only covering a few dozen genes? Why don't you get as much information as you can with whole genome? The problem is that it's a trade-off. More information usually takes more time and usually requires better samples. And obviously for cholangiocarcinoma and biliary cancers, those good samples aren't necessarily available. So one of the big jobs that we as pathologists have to do is to strike the right balance between these competing demands. Get as much information as we can while making sure that we do actually get information and the test doesn't just fail. And so mostly what we do nowadays is probably the medium-sized panels, but increasingly the large panels as the technology gets better. This is an example of what you might get on a small NGS panel. So there are a few dozen genes on here. And it's tempting to look at this and think, well, if a cancer is tested with this panel, any mutation in any of these genes will be detected. That's not necessarily true, though, and I think this is an important point to recognize. Let's imagine that we've got a biliary tract cancer and there's a BRAF mutation. BRAF is covered by the panel. You would quite rightly assume that if there's a BRAF mutation there, it would be in the report. But an important thing to recognize is that most of the time, the scientists in the genomic laboratories will only look at the genes which are clinically relevant to the disease in question. This has changed a little bit in the last couple of years, but historically that would have been IDH1, meaning that although there is a BRAF mutation there and although it was covered by the panel, it won't necessarily make its way into the final report. And it's important to recognize that because you could potentially think there isn't anything there. Actually, it just hasn't been looked at. And I'll come back to why this is important in a moment. So that's genomic testing. That's looking for alterations at the level of DNA and RNA. What about proteomic testing? So looking at which proteins the tumor cells are expressing. Well, there's really only one way of doing this in routine practice at the moment, which is immunohistochemistry, which I mentioned at the beginning. The idea here is that you have cells expressing particular molecules. So this is HER2, for example, expressed in the cell membrane. We in pathology apply an antibody which binds to that HER2 protein, and that antibody has attached to it a colored molecule. It's a little bit more complicated than that, but fundamentally that's how it works. And that means that when we look at the tissue down the microscope after it's had immunohistochemical staining, we see cells which are expressing that molecule as looking brown. So you can see there, we've got, this is actually a cholangiocarcinoma stain for HER2, and the brown lines around the cells indicate that those tumor cells are expressing the HER2 protein. Now, this used to be really easy for us, because in the past, we would just say brown or not brown, positive, negative. Things have become more complex, though. Increasingly, we're having to do quantification of these biomarkers. So how many cells are brown? How brown are they? How much of that brown goes around the cell membrane? And it's getting to the point now where this is actually pushing against the limit of what a human can do unaided. And this, I think, really does raise the need that we digitize this and get this done by algorithms which can actually do it reliably. So that's a lot of the theory. Let's snap back to the practical now. What does this mean for delivering molecular testing in biliary tract cancers? These are the standard targets that we look for. And I stress this is my opinion. If you want to know about IDH1, I actually think that from the perspective of IDH1 alone, NGS or a single gene approach is pretty good. They will pick up the alterations. Obviously, if you want to know about things other than IDH1, NGS is your best bet. But I think if it's just IDH1, either of those is fine. FGFR2, NGS is the winner. It is clearly superior to other approaches. Likewise, NTRK, it's a little bit more controversial here, but I strongly think that if you want to know about NTRK, put it on an NGS panel. For MMR, MSI, you genuinely have a choice between a proteomic and a genomic approach. The weight of evidence to me suggests at the moment that MMR is still probably superior, although I suspect that NGS is probably just as good. We just need more evidence. For HER2, the currently approved drug, its license says that it has to be by immunohistochemistry, therefore it's proteomic. And the point I want to make here is that even for cholangiocarcinoma, which historically been a very genomic-led disease, we now have nearly half of the biomarkers being proteomic, and this comes back to my point about proteomic testing being the poor relation of genomic testing. Proteomics is important, and I'll return to that in a moment. So, with all of that, How are we actually delivering these tests in different parts of the UK? And I've got to say here that most of this work didn't come from me. It came from colleagues, some of whom are in this room, who've provided the information about the testing situation in the various nations of the UK. So first of all, the lay of the land. It's in pathology departments that the diagnosis of cholangiocarcinoma will be made and where the proteomic testing, so the immunohistochemistry, will be done. It's actually remarkably difficult to find out how many pathology departments there are in the UK, but that is my best attempt from the research that I did. The answer is lots of them. It's a very, very distributed system, and that in itself can cause issues. If you look at the genomic laboratories, the kind of official commissioned ones, the number is much, much smaller, and this number has actually decreased in recent years. In England, the English Genomic Medicine Service comprises 7 genomic laboratory hubs with associated smaller laboratories. The number of those is decreasing as they consolidate. In Northern Ireland, there's a single laboratory in Belfast. In Scotland, 4 regional centres delivering testing, and in Wales, a single consolidated laboratory in Cardiff. Now, I should say there are other providers here. There are private providers, there are NHS non-nationally commissioned providers, but this is broadly the lay of the land. So you have a large number of pathology departments feeding into a small number of genomic laboratories. So, the first question I'm going to pose is, can patients in all parts of the UK access the standard of care target testing? And that would be the testing for these 4 markers which I've described here. And can they access that by NGS? And the answer is yes. Thanks in large part to many of the people in this room, and particularly to the AMMF. And I think it's easy to undersell how big an achievement this is. You know, it is actually very difficult to roll out this testing, and the fact that this is available in all nations of the UK is a phenomenal achievement. In addition, in England and Scotland, there is also access to second-line single-gene approaches if NGS fails. That is not available in all parts of the UK, though. Second question, who pays? The problem here is that if it's the requester who ends up paying, so if I as a pathologist request a test and then the bill comes back to my pathology department, there's a disincentive there for me to request, because the more we spend on genomic testing, the less I can spend on scientists in the lab to help get the work through. So that's a problem. And therefore, I think it's a very good thing that this is centrally funded. The bill doesn't come back to me. It gets paid, obviously, but I'm not the one who has sight of that, and that means that there isn't a disincentive for me to request the testing. Again, this has been very, very hard won. Third question, who is eligible for this testing in each part of the UK? And this is where things start to differ, because in England and Wales, that is the official line. Now, in reality, nobody is actually policing this. But to me, there is a problem with that, in that it implies that you should only be initiating testing at the point that the patient is being considered for second-line treatment. Whereas I would argue that if you're waiting until then, it's going to be a huge rush, you're going to end up delaying downstream decision-making. This should be done early in the process. That also helps with diagnosis, as I mentioned earlier. So I think work needs to be done here, and in fact, this is being looked at by NHS England. In Northern Ireland and Scotland, they have their own, I would say, more pragmatic approaches, which have been developed by the clinical teams there and which I think are sensible. Fourth question, because I've already explained that across the UK, we have really, really good provision of those core biomarkers which are essential to treatment. But do the genomic laboratories issue any additional results alongside those? Because that's the big benefit of NGS. You look at all of these genes, it seems a shame that all that data just sits on a server and doesn't go into a report. A lot of progress has been made here. So in England, you will also get information about these non-standard clinical trial targets. And in Scotland, there is also a similar list, but I would say it's probably a bit more curated towards biliary tract cancer. In Northern Ireland and Wales, these things are not reported routinely, and I would say this is an issue. And it's an issue which is about to get bigger, because England is currently rolling out these additional targets on top of the previous ones. Now, as I'm going to mention later, I don't think that's a reason to hold back England's progress, but it is a gulf which is emerging between the nations, and I think that's problematic. Now, that's all about the genomic stuff. What about the proteomics? in a way, the good news is that we do have equity, but I would say that equity is not in a good way, in that I don't think the system for delivering proteomic testing across the UK is particularly robust. Any laboratory can choose or not choose to deliver proteomic testing, and there is no rule book that says that patients with, say, biliary tract cancer must have HER2 testing. There's no obligation in the same way that there is genomic testing. There are no formal referral pathways. If you want to send a case for HER2 immunohistochemistry, you just find a lab and you send to them. Now, Birmingham, we do about 75% of the country's work for this, which I'm not complaining about. We enjoy doing the work, but I don't think that's a particularly robust system nationally. It doesn't give us very much resilience, and I think we need more centres doing this testing so it's spread out. And most importantly, the funding arrangements are extremely heterogeneous. So the funding is generally local, meaning that the pathologist who requests the test gets landed with the bill. And that is a problem because it disincentivizes requesting. So, summary, I think we can all agree that all patients should be able to access high-quality genomic and proteomic testing to facilitate access to standard and increasingly clinical trial targets, and no patient should be disadvantaged by their postcode. I think that enormous progress has been made in getting access to the standard genomic targets, and that isn't something which should be downplayed. But I think we're seeing an emerging gulf in those extra targets, and what I think we shouldn't be doing is suppressing the services like those in England, which are delivering more testing. Instead, and I appreciate this isn't an easy thing to do, we should be levelling up all 4 countries of the UK so that we're all delivering that very expansive repertoire. I think that we need to move away from eligibility that says you get tested when you're being considered for a treatment to a system which promotes testing earlier in the pathway. And these are my words, not the words of NHS England. Proteomic testing, in my opinion, in the UK is a mess. And I think that needs to be worked on in the same way that genomic testing has been worked on. We've done it for genomic testing, and there's no reason we can't do it for proteomic testing as well. So thank you very much for your attention.